Hello, NEB. My name is Michael Doherty. I'm a second-year graduate student in Northeastern University's Cell and Molecular Biology program. My lab studies the genetic regulation of cell migration using the nematode worm C. elegans. On the day-to-day, we do a lot of molecular cloning in my lab. Having said, we rely on high-quality DNA polymerases to assure that our clones are made exactly as we designed them. We've been using your Fusion hot start polymerase for some time now, and we've been very impressed with both its fidelity and speed. To test the new Q5 polymerase, I designed a side-by-side study with Fusion polymerase to see which enzyme could better amplify a short region of plasmid DNA.
Alright, so what I hope to do here is amplify a 600 base pair fragment of this plasmid PREP NTAP. That 600 base pair fragment actually codes for a tag that I hope to use in some of my protein purification experiments. So this tag here, the N-terminal tap construct, is composed of an IgG binding unit domain, a TV protease site, and a calmagulin binding peptide. This tag is going to be used for some of my proteomic studies and is very effective in purifying proteins nearly at 100% because it eliminates unwanted contaminants by using two different modes of purification. In this cloning project, I hope to recombine this insert with the vector that I currently have by using infusion ligation. Infusion ligation is different than classical ligation because it does not use a ligase like T4 ligase. It uses a viral recombinase enzyme. That viral recombinase enzyme has different requirements for how the recombination of the two DNA molecule works. It requires that primers that have 5' overhangs complementary to the vector are present. So this means that for the first couple cycles of PCR, your primer is not going to completely anneal to your template molecule. This can make this sort of amplification more difficult, and this is why I chose to use this sort of system to test the Q5 polymerase against fusion. After performing PCR with the infusion-based primers, what you're going to be left with is an amplicon that consists of your amplified area of interest, plus on both ends of the molecule 15 bases of complementarity to the vector in which it's going to be cloned into. With all that aside, I'd like to show you how I set up these reactions side by side. One of the first things you see when looking at these reaction setups is that the Q5 reaction uses half as much enzyme as the fusion reaction. This is advantageous because as these enzymes are both formulated at 2 units per microliter, you're going through that stock of yours at half the rate as you are the fusion reaction. This may save your lab or institution quite a bit of money in the long run. The only other clear differences here between the two reactions are the buffers in which the enzymes are formulated, and that I decided to add the 5x GC enhancer to the Q5 polymerase reaction. Other than that, the concentrations of all the other reagents are the same, and the reaction volumes of each reaction are the same, 25 microliters. Based on data provided by the product sheets, I was able to come to the decision that I would use the same PCR method to perform both of these reactions. If you take a look at the method that I'm using, there are a couple of highlights that I'd like to point out. One is the annealing temperature of 51 degrees C. This annealing temperature is set at 51 degrees because it's 5 degrees less than the melting temperature of the lower of the two primers that I'm using. The other thing is that I'm using 40 cycles of PCR as opposed to the 25 to 35 recommended cycles. I'm using 40 cycles just to maximize the amount of DNA yield that is produced from this reaction. So after pressing run on my thermal cycler, I made note that these reactions were only going to take an hour and a half total. That was great to see, as PCR can be quite a lengthy process. It's always great to save time and provide more opportunity in your day to perform other experiments. After my PCR cycle was finished, I proceeded to load these samples into a 2% agarose gel. So what we see here are the bands of my NTAP amplicon at 646 base pairs. And if you look from left to right, you see the fusion polymerase, the Q5 polymerase, and the Q5 hot start polymerase. So I did test both of the enzymes that were provided in the free sample. And it's difficult to see here, but I do believe that the two Q5 polymerase bands are substantially larger than the one produced in the fusion polymerase, thus resulting in more DNA products. So one thing that I did not do is check the fidelity of these enzymes. So I did not excise these bands and send them from sequencing and test them against their template. But one thing that I can say is that for half the price per unit of enzyme, I would highly recommend using Q5 polymerase for standard cloning procedures. The yield is excellent, and it does not seem to produce any sort of misprimed amplicons. I'm quite happy with this free sample, and will most likely proceed to purchase it in the future.
Lastly, I'd like to thank Northeastern University, the NIH, and of course NEB for pushing my research forward every day. Oh yeah, one more thing. This presentation would have been a lot better if it was made on a Mac.